So hi everyone, today our group will be explaining the helminth infection for Necator americanus. So let's see what is Necator americanus. So Necator americanus, which also known as American or New World hookworm, is a species of parasitic intestinal hookworm. The shape of this hookworm is cylindrical and slightly curved. Both male and female adult worms have buccal capsule with cutting plates for feeding, as you can see in the figure 1 and figure 2. Now, let's see Necator americanus egg. This egg appears as oval-shaped and thin-shelled, and it is roughly 64 to 76 micrometer long. It cannot be differentiated microscopically from Ankylostoma diurnal eggs, as you can see in the picture. Necator americanus can be found in areas with poor sanitation and also soil which is in warm, humid, and tropical regions of Central and South America, Caribbean, Africa, and parts of Asia.
So this is how Necator americanus infect host and its life cycle summarized in 5 stages. The first stage is the eggs which are passed in the stew. Then the second stage, the eggs hatch into reptidiform larva. Then the reptidiform larva develop into fidariform larva, which is infective. The next stage, fidariform larva penetrates skin of human host, enters blood circulation where it's carried into the heart and then the lungs. The final stage, the fidariform larva is coughed up, swallowed, and ends up in jejunum or small intestine, where the larvae develop into mature adult worms. The adult worms attach to small intestine wall, causing the host blood loss, and the adult worms lay eggs which are passed in the stool, repeating the cycle again. So there are two forms of Necator americanus larva. As you can see, both forms have pointed tail and striated sheath, and the reptidiform or in non-infective form has a large bulbous esophagus. Necator americanus infection is a serious problem shown by its epidemiology. In the USA, a 2007 study states that 95% of human hookworm infections are caused by Necator americanus, and in China, a 2021 study states that from 25,631 cases of hookworm infection in Henan province from 1949 to 2020, 2,596 cases were caused by Necator americanus.
So I'll be explaining about specimen collection and processing. Necator americanus is diagnosed by identifying its eggs in a fresh stool sample. To collect this specimen, we use a clean, dry, wide mouth container, taking care to avoid any contamination with urine or water, as this can interfere with egg visibility. It's best to collect the sample in the morning since egg output is highest at that time. For processing, a direct wet mount using saline or iodine can be done for quick egg detection. However, to improve diagnostic sensitivity, concentration techniques like formalin ether sedimentation are recommended. If immediate examination isn't possible, the stool sample should be preserved in 10% formalin. This helps maintain the structure of the eggs for later analysis.
Next, I'll be explaining the advanced diagnostic techniques for Necator americanus. The first one is PCR, which is polymerase chain reaction. This helps to detect Necator DNA from stool samples, and this also is a method that is highly sensitive and specific for detection of Necator americanus infection. The next one is real-time PCR, which quantifies DNA for the evaluation of infection burden from stool samples. The third one is ELISA, which is enzyme-linked immunosorbent assay, which is the diagnostic instrument used to detect antibodies in the bloodstream that indicates a current or previous infection with this hookworm. And the last one is LAMP, which is the loop-mediated isothermal amplification, which is less complex than PCR, and at a high constant temperature, it amplifies differently.
So today I will be explaining the modern versus conventional techniques of diagnostics. Modern techniques involve the use of PCR and molecular sampling. They offer higher sensitivity, more specificity, can be used to differentiate species, provides rapid and quick results, and is but the only downsides are they're very expensive and require a high level of technical expertise, and most methods are yet to be validated. The conventional techniques involve observation of stool samples, and these techniques are simple and inexpensive. They are very readily available, and they give insight into the estimation of infection or the phase of infection. But the only downsides are that they have low sensitivity and require expert level of understanding of the larvae to diagnose.
So, in conclusion, helminth infection remain a public health concern, especially in unreserved regions. Effective control relies on understanding the epidemiology and ensuring proper specimen collection and processing. While conventional diagnostic methods like microscopy are still used, advanced techniques offer greater accuracy and early detection.